I'm Dr. David Rubin from the University of Chicago Medicine. Welcome to Best of the Day from the Advances in IBD meeting in Orlando, Florida. This is the National Crohn's and Colitis Foundation of America meeting, and this is the 13th annual meeting, although the first time we're broadcasting and having the meeting in Orlando. The meeting's running from December 4th through the 6th and highlights both the clinical and basic science and clinical management updates throughout our field. I'll be hosting a number of these interviews, but my first interview today is with Professor Maria Abreu, one of our colleagues who is also the division chief of the Division of Gastroenterology at the University of Miami. Maria is on the planning committee for the basic science component of this important meeting. Welcome, Maria.
Hi, David. It's great to be here.
It's first time in Orlando, a little further from where you are in Miami.
A little bit further, but a short flight, so yes. I can't complain.
And the weather is still very nice.
Still very nice.
Yeah.
Still very nice. I wanted to begin by telling you that I had a role to play, as you mentioned, in planning the basic science part of the meeting, and I think that one of the things we really are trying to strive for is having this be the cutting edge of IBD research being presented here. We realize there are lots of meetings that we're competing against where science is presented, but it's amazing to me how over the last few years I can see with every year the quality of the abstracts, the quality of the presentations keeps getting better, and it's awesome. In looking through the abstracts and just sort of in no particular order, but just to give you some of the things that I think are really kind of interesting that are happening. In genetics, we've gone sort of in a pendulum sort of way. We first started by looking when we had clunkier ways to try to identify genes. People would sort of sit around and think about a gene and think, God, TNF must be important. Let's look at every mutation that there could be in TNF, to pick an example. Then the next wave, when we became sophisticated at just sequencing everything and being less biased, we discovered not to, right? And then at a higher macro level, we started doing genome-wide association studies and sort of identifying genes, but more regions, right? Because, as you well know, we don't even know which of these genes are really causally involved, or if it's a neighboring gene, or if it's even sort of a mutation in a different part of that gene. So we went in that direction, looking at these enormous populations and then doing meta-analyses on these large populations and tens of thousands of people with IBD. Now, I would say that we're going a little bit, not in an opposite direction, but rather in a much more targeted way. And there's a lot of emphasis in the genetics world on almost like autosomal dominant conditions, and this very early onset IBD that Alexis Musi from Sick Kids and Scott Snapper at Harvard have been pioneering. And it gives them the opportunity, you figure that if children are presenting with severe forms of IBD at a very young age, that they must have a very high genetic susceptibility. They've identified some things that are almost autosomal dominant, like the IL-10 receptor mutations. At this meeting, they'll present data that they now were trying to identify more like autosomal recessive reasons for having IBD. And the way they did that was twofold. One was looking at particular genes and seeing if there were either two of the same mutation, or what are called compound heterozygotes, right, where it's the same gene is mutated but in two different places, so in the composite the gene is not working. Mm-hmm. There are people who, for example, are compound heterozygotes of the NOD2 mutation, as one example. And so using that approach, as opposed to sort of purely an autosomal dominant approach, they identified that there were kids that had mutations in RAG2. RAG stands for recombinant activating gene, which is the gene that slices and dices T cell receptors and B cell receptors, aka the immunoglobulin genes, to provide them specificity. So we use RAG knockout mice, for when we wanted to use immunodeficient mice as an example. And so these immunodeficient pathways that are defective in B cell signaling and T cell signaling seem to be a theme. They found other kind of signaling pathways that were defective. They also used another approach that I think is another thing that is that we've now incorporated into the field, which is kind of systems biology approaches, not just looking at a gene but at a pathway. So they wanted to see if they could find kids where there were multiple mutations in a pathway that was causing this autosomal recessive characteristic.
So when you talk about autosomal dominant and recessive, it's not the same as talking about inheritance patterns, is it? Or is it just the way the genes are expressed? Is it more of the penetrance?
Yeah, the penetrance. I suppose that the classical Mendelian way of thinking about things is challenged in a disease where we know there has to be an environmental trigger and other factors. And so, you know, in my mind, an autosomal dominant disease is that even when you have one bad copy of the gene, either that bad copy can superimpose its function on the good copy of the gene, because it can mess up the entire signaling cascade. You know, or in the case of something that is autosomal recessive, that just one copy in one gene, the other good gene is able to make up that function. And so, how that in the context of IBD is playing a role, I think, you know, the IL-10 receptor story is a clean one, as clean as it could be, right? Because there's really very compelling data in vitro studies and even bone marrow transplants, right, for wild type.
And
IL-10.
just for the audience, those were infants, basically. I mean, 2 years or younger when they got these very obvious expression of a terrible IBD. And just to point out as well, you're saying IBD, you're not saying Crohn's, UC. I mean, when we talk about these things now, I think that's appropriate. We need to be stepping back and just acknowledging that we're dealing with lots of different diseases. Yeah,
I think that that's a very astute point. And that in children, sometimes they have colonic involvement, but it's very clearly not what we would, you and I as adult gastroenterologists, think typically about ulcerative colitis. The other, you know, the other things, another kind of cute genetic story that caught my eye because of the methodology was a study that has come from Mount Sinai, actually, that looked at looking for genetic causes of collagenous colitis. I normally don't think about or conceptualize these diseases as genetic, but who knows exactly? And what they did was, you know, collagenous colitis isn't that common. I mean, it's common. We all have it, but you don't have a clinic full of them, right? You see them, I'm assuming, like I do, which is you saw mostly IBD that day, and then there's a person that comes to see with collagenous colitis, making it hard because there are not so many of them. So what they did was they pulled together the microscopic slides, and they isolated the DNA from the formalin-fixed slides.
Cool.
And then they used that, so they had, I forget, hundreds of patients that way in the Mount Sinai, you know, pathology library. And then they compared that against the control data. I think the controls were really kind of Crohn's and UC from the IBD consortium that they had available to them. And they actually found some genes that were common, and that doesn't surprise me, right? I mean, you have a disease that causes intestinal inflammation, there are probably some commonalities in those pathways. But they also found some signals that suggest that it could be CD4 T cells that, you know, because it's like class 2 MHC, you know, the usual, like in UC, but different in different parts of the MHC region. So I thought that was kind of cute. We have, I think the other thing that we're getting a little bit away from that, in fact, kind of glancing through the oral abstracts, I didn't see any real, like, okay, we did another microbiome analysis of this mouse or of even of this human or of these Crohn's versus UC. You know how we had a blip of that kind of stuff? I think we now recognize that these names of bugs is probably not all that important, but that really the harder part will be, what are they making? What are they actually doing? Like, what are they synthesizing or not synthesizing that is making people prone to developing IBD? We had a paper that our group is presenting, and, you know, it got picked. I didn't have anything to do with it, so I don't want to have the audience think it was biased. And it kind of conceptualizes a story. We made a mouse that's prone to developing inflammation, but if you do nothing and nothing happens, if you don't do anything to the mouse is revved up in its ability to recognize bacteria. So we figured there must be a change in their microbiome of these mice, and there is some change. But the really neat part of the story is that what it does to change the microbiome, that just making that one change in the lining epithelial cells, can be transmitted to another mouse through the microbiome and make those mice then susceptible. Do you know what I mean?
Yep.
You didn't transfer the genes.
I read your abstract.
Well, get out!
I did.
Someone read the abstract.
I read your abstract. No, so I think that the whole idea of understanding a little bit more about how not only do we look at what's in the gut, but how it's being regulated or the interaction is key to all this.
Exactly.
And I thought that was really clever.
So that mirror image thing, there's the host side, but then there's the microbiome side, and it's an iterative process. And that's, I think, part of the reason we need to do something like the GEM study that initiated in Canada, but now is being done more broadly.
So the GEM study is looking at unaffected siblings of people who have IBD to try and follow them longitudinally and understand risk factors for the development of new onset IBD.
Right.
So that is exciting. So talk to me a little more about the microbiome and some of the other things that will be presented at this meeting.
The other things actually that I thought I'd mention to you, David, because I also saw that there was a bit of a theme that people are now doing stuff and doing experiments that are much more translational. Some that are, for example, things like trying to identify small molecules that will have an alpha-4 beta-7 inhibitory effect. And I think that's kind of interesting because actually the particular abstract, one of the orals that'll be presented, they gave it orally. And they talk about inhibiting trafficking of alpha-4 beta-7 T cells to mesenteric lymph nodes. And so you start thinking about, we've been conceptualizing, or at least I have been conceptualizing, that you needed to have it be coming from the systemic circulation, that this was going to block trafficking just at that level. Mm-hmm. And it probably still is, but at a very micro level in the microcirculation in the gut. And so that's kind of cool because if that can be proven in adults, then you can start thinking about, lessening side effects and all the consequences of maybe systemic exposure.
So a bit more targeted.
More targeted. There was another one that if you were looking at the abstracts that looked at lymphotoxin beta receptor signaling in epithelial cells of the gut. Why is that interesting to me? This lymphotoxin receptor, which is in a TNF receptor family, they found it on epithelial cells, fine. On epithelial cells, it appears to trigger IL-23 production. Okay, fine. IL-23 is important as a cytokine. It's been shown in all the various genetic studies to be important. But IL-23 leads to the activation of innate lymphoid cells in the lining that go on to produce cytokines. IL-22 is one of them, but who cares about the name? That serves a cytoprotective function, so it protects the lining epithelium. So what I thought was interesting about that was a couple-fold. One was that on the one hand, remember that etanercept doesn't seem to work in IBD, and that blocks lymphotoxin signaling. And I thought, well, I wonder if it's because it's sort of blocking this protective pathway inadvertently. And then finally, of course, we know that famously the IL-17 antagonist studies were not effective, and not only not effective, but in certain cases actually caused an exacerbation of Crohn's. And so you can think through that perhaps it's having this inhibitory effect on this beneficial pathway. So it's always tricky, right, to kind of tease these things apart.
How close do you think we are to really customizing therapies based on some physiologic assessment or pathophysiologic assessment of an individual patient?
Yeah.
I mean, we have all these clues in place. We have more therapies coming that are expensive.
Yeah.
Just from a translational point of view, since you brought it up, when do you think we'll start looking at those markers so that we can choose the right therapy and avoid all the time and expense of just going down the line alphabetically?
Yeah. It's sort of trite to say that I think we're closer than ever. Becomes a much cooler issue now that we have other mechanisms of action that we can actually reasonably have, deploy for patients. And I think that so far as I know, we haven't identified markers that will predict response to something like vedolizumab, an alpha-4 beta-7 antagonist. I think that because we need to now reshift how we think about it in that, since we're inhibiting trafficking of certain T cells to the gut, what we need to know is what is it that we've kept out that we're having a beneficial effect by keeping certain things out, and maybe some other things not out.
Right.
And so I think we have to kind of reframe how we think about these things. I also think that the other step that we need to take is that a lot of the studies so far that have looked at even what's expressed in the mucosa and things like that to predict response, have been what I call find and grind experiments. It's taken everything in a piece of tissue. And you and I can talk to you about science, but we're endoscopists, right? We're taking a biopsy, and sometimes we're biopsying an ulcer, and sometimes we're biopsying near an ulcer, and those are different. That has different proportions of cells and things in that tissue. So we need to deconvolute either in a computerized way, in silico way, deconvolute what's in that tissue. Or I think, actually, as a purist, that we need to sort of separate different cell populations to see exactly what we're doing in these individual patients. And so I think, actually, in the face of having something as potent as anti-TNF still are, and my bias is that they're safe medications, I think what we need to figure out is the few that don't respond, kind of why, or in those that don't fully respond, what's the complement to that? Mm-hmm. Like what is keeping that last bit of inflammation around, right? Pharmacokinetics aside, the cool thing would be, what is that delta? What is being made up? Sure. What is the bypass tract? And so I think that I don't have the answer to that, but I think that's where we as a field should start thinking about.
So I saw a statistic yesterday that the CCFA this year is expected to invest $20 to $22 million in the next year alone in research. And when we see all these advances, and we're hearing about all this here, I think that's a really nice number. But what is the national scene for research dollars and funding?
Yeah.
What is your take on all that, just so people out there have a sense for where this is going and why they should make sure they're supporting these efforts?
Yeah. Well, I think that as you well know, at a national level, there's been a very dramatic reduction in the availability of funds for any number of reasons, geopolitical reasons, really. And what that has encouraged, much to my dismay, is very incremental research, because it's the big labs doing very safe things, or at least taking the money and doing what they want, but proposing to do very safe things, and that gets funded. And I find that objectionable, because I think some of the greatest discoveries that are made really have nothing to do with the quotidian, with the things that are expected. The really cool things are the unexpected and the crazy ideas. Yeah. And so I think that the CCFA, in combination with, I think, the Broad, now that they've, as you know, the Broad is going to roll their resources also with the CCFA to continue to fund edgy science. I mean, that's what I would consider it, edgy science. And of course, I think the CCFA group as a whole, it's phenomenal, the proportion of their money that they invest purely in research. They have been very strategic in a very good way. I mean, a lot of these efforts that I've described, microbiome efforts, the genetics efforts, they really have pushed and even gotten nationally the NIH to also kind of subsidize research that is beneficial for our community of doctors and patients. So I think the CCFA, to me, is still it's our family, it's our core organization, because I think they've really made the most strides for patients and for the research in IBD.
Yeah. Well, we've talked about a lot, and that included all the updates that we're going to hear about, some genetics and genomics at this meeting, and some microbiome. But what's exciting to me, and I think to the people who are watching this, will be how we're starting to tie things together, how we're looking at both the microbiome, the genetics, and now epithelial cell biology and regulatory pathways to finally start making a story out of all this, as opposed to a laundry list of discoveries that didn't have any link. So that's great. And so thanks for your leadership and efforts on all this here.
Thank you, David.
And I hope that the rest of the meeting is a good one for you.
Same to you. Thank you.
So I'm David Rubin, and joining me was Maria Abreu, the division chief from the University of Miami, and we're at the Advances in IBD meeting in Orlando, Florida. Thanks for joining us for Best of the Day.